So we're looking at the all too common slide of blood. Yeah, so this is a blood smear, which you've looked at in the past, hopefully in biology or in anatomy. So what you're looking at there is, if you recall, well, you are seeing red blood cells. So here are a lot of red blood cells, or otherwise known as erythrocytes. Those are not necessarily part of the immune system. What is important for the immune system is these cells here. Okay, so these are called lymphocytes. Lymphocytes are important because they do provide protection against pathogens, whereas red blood cells, well, they are pretty important. They're important for gas exchange and gas transport and pH stability in the blood. They deal with oxygen and CO2, but they do not kill bacteria. They don't deal with viruses, whereas lymphocytes do. Okay, so we have two major types of lymphocytes that we're going to look at in this lecture. We have B cells and T cells. Okay, so B cells have an origin within the bone marrow. Okay, specifically yellow bone marrow within, evidently, the medullary cavity of long bones. Okay, if you're not familiar with that, I suggest looking up some videos, reading through your anatomy textbook, or look it up online. Medullary cavities of bones, they contain bone marrow, two types. One of those types contains lymphocytes, not necessarily lymphocytes directly, but the stem cells that give rise to lymphocytes. They produce lymphocytes. Then they travel immature over to the lymph nodes, and within the lymph nodes, these lymphocytes manage to generate mature B cells, which have the capability of producing antibodies and undergo clonal selection, which I'll go over in a few minutes. The other type of lymphocyte is T lymphocyte, or T cells. These are generated again in the bone marrow, but they travel to the thymus, to the thymus gland, which is located in the thoracic cavity, specifically in the mediastinum, right above the heart. So if you want to know more precise location, then go ahead and find the middle of your collarbone, your sternum. So right behind it is the location of the thymus. The thymus gland is important for T cell maturation. Okay, so T cells, even though they're not produced, they're not generated in the thymus, they're generated in the bone marrow, but they travel to the thymus to be able to mature and be able to produce the two major types of cells that T cells divide up into. Okay, they divide up into helper T cells and cytotoxic T cells, which you've probably heard about in the incidence of HIV. Okay, helper T cells are important in HIV pathogenesis. Okay, so an important function of each one. Helper T cells are crucial for or within cytokine signaling. Cytokines are, for the most part, signaling molecules that induce and call on the immune system in case pathogens are present in the body. And then cytotoxic T cells, well, they attack infected cells by either bacteria or viruses. So, sorry, let me go back here. There's a few attributes to the adaptive immune system, and these are going to be important for the functioning of the adaptive immune system. These are specificity, inducibility, clonality, unresponsiveness to self, and the production of memory cells specifically. So what's important here is that adaptive immune cells are very specific to pathogens. Okay, so they're not going to act in a broad or general manner like cells within the innate immune response. They are going to be very specific to antigens, but regardless, collectively, all of your B cells and T cells, in a collective manner, those can attack a very wide range of viruses and bacteria. Okay, so it's said that that they're specific for millions and millions of pathogens and bacteria that that attack our body, within different points of our lifetime. Inducibility is another characteristic. Important characteristic of the adaptive immune system. It refers to the fact that adaptive immune cells cannot work in and of themselves. They have to be induced by signaling molecules called cytokines. Okay, so I'll go into details on that. Clonality is another attribute, and memory is another attribute. Clonality refers to making lots of copies, so identical copies of B and T cells, and in a process called clonal selection. And then memory is once you have a good set of B and T cells produced, then half of those go towards building memory in case you're exposed to the same pathogen in the future. And then the rest of those memory cells are actually they're on alert essentially all the time. So here's a bit of an overview of the adaptive immune system. It's divided up into two branches, into a further two branches called the cell-mediated immunity and antibody-mediated immunity. Okay, so again, here's a bit of an overview before I get into the nuts and bolts of each.
Host defenses consist of two categories, the innate or non-specific defenses and the adaptive or specific defenses. The human body has many innate defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system. The human body has two main divisions of adaptive defense: humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The adaptive immune response is divided into two categories: humoral immunity and cell-mediated immunity. A humoral immune response consists of the activity of B cells and the antibodies they produce.
Again, two divisions to the adaptive immune system. One is humoral immunity, which is an antibody-mediated immunity, meaning that our cells are going to go ahead and produce antibodies, mass produce them, and then those antibodies are going to go ahead and find the pathogen that is infecting the body to be able to deal with it in several manners, which I'll cover in a few minutes. And then the other branch is a cell-mediated immunity, which utilizes B cells to be able to again mass produce antibodies, but deal with pathogens in a different manner, which is very specific in order to elicit the destruction of those pathogens. Here's an overview of the lymphatic system. It's important to go over the lymphatic system because it is in charge of maturing B and T lymphocytes, and it does allow them to mature and gives them their ability to produce antibodies. Okay, the lymphatic system is made up of a few organs. First is glands. Okay, so there's glands, for example, the thymus gland, and there's another type of tissue called MALT tissue, or mucosa associated lymphatic tissue, which includes the tonsils and nodes, and then what's called Peyer's patches. These nodes are located, they're concentrated in several different regions throughout the body, to include cervical. These are cervical nodes right here. Cervical refers to neck region. There's also axillary lymph nodes, which are located in the armpit region on both sides. We also have inguinal lymph nodes, which are located in the groin area. There's also Peyer's patches, and Peyer's patches are located in the abdominal region within and outside the intestines. Okay, the spleen is also involved in the lymphatic system. The thymus gland is also involved in maturing T cells. Lymph nodes mature B cells, and everything else contains and circulates, for that matter, B and T cells, and allows for them to circulate and work by looking for pathogens, attaching to them, and destroying or helping destroying them. Okay, another feature of the lymphatic system that is important here is the lymphatic vessels. Okay, lymphatic vessels, they're very similar in some way to veins. Okay, and they're similar to veins because they work in, there's flow in one way. Okay, so veins and arteries work in that manner. They also have valves. Okay, so in case there's nothing to push this lymph fluid, which is located inside the vessels, for example, up from the legs, then these valves are going to go ahead and be in charge of allowing, and you're looking at a valve right there, they're going to go ahead and allow for lymph to move upwards in a rhythmic manner, and they're not going to allow for backflow of lymph fluid, which is especially important in the legs. Okay, so any buildup of lymph fluid can cause edema or inflammation. All right, so lymph fluid is generated from the percolation of blood plasma. So this blood plasma that is accumulated in the tissues normally it goes ahead and is filtered right through the walls of the lymphatic vessels, and at that point it's not plasma anymore, it's not blood anymore, it becomes lymph. And lymph is a clearish yellowish fluid which contains a lot of B and T cells and antibodies, which are important in the destroying bacteria, as you know. Here you're looking at a cross-section of a lymph node. Notice that the lymph nodes have several regions of importance. First is a medulla. Medulla is the middle region of the lymph node. Cortex is the outside region, so the outside of it, the most superficial aspect, is called the cortex. There's also vessels that lead into and out of the lymph node, and these vessels are characterized by the fact that they have valves to prevent backflow of lymph. Why are lymph nodes important? Well, because they act as filtering systems for plasma. Okay, so and blood for that matter. So they filter blood and plasma by using lymph fluid, and they help blood get rid of microorganisms that might be infecting the body. So antigens are molecules that are generally located on the cell surface or the cell membrane of anything that is made up of cells. Okay, you know, whether it's a eukaryotic organism, a prokaryotic organism. Eukaryotes contain a nucleus. They contain antigens. Our bodies, our cells are full with antigens. But let me define it real quick what antigens are. Antigens are molecules that provoke specific immune responses. Okay, so they're going to go ahead and trigger the immune system to act against them. Antigens contain epitopes, which are also known as antigenic determinants. So these are specific regions on the outside or located on the outside of the cell membrane on the antigen itself. And these regions are specific for antibodies. Okay, so antibodies go ahead and bind to these regions. And say, for example, that this antigen belonged to a bacteria, then the antibody would go ahead and bind to the epitopes, and it would go ahead and either neutralize or opsonize that bacteria right away. From there, it would go ahead and take a piece of the antigen, and then it would process it either endogenously or exogenously. In the case of a bacteria, it would be exogenously, because these are pathogens that come from the outside, and that would essentially build what is known as an antigen presenting cell, which in and of itself triggers a very specific immune response by using the adaptive immune system and also the cell-mediated immune response. Okay, so even though most of our cells are nucleated, with the exception of red blood cells, most of our cells contain antigens. Okay, but these antigens are actually recognized by all of our antibodies, which means that they won't trigger immune responses. Okay, because again, we were born with them. We recognize them as not foreign, as belonging to our cells. But antigens can also be found. They don't have to be found on cells. They can also be found in protein products of viruses, of bacteria, fungi, protozoa, and basically any microorganism that can cause disease will fundamentally release some sort of antigen that will trigger, once inside of a body, it will trigger an immune response of the host organism. It's important to note that food and dust can also contain antigens or allergens. Yeah, so depending on whether somebody's allergic or not, those can become allergens, which contain antigens that trigger, again, that immune response, which allergic people are known for, essentially. Alright, so these are the different sources of antigens. There are three general sources: exogenous, endogenous, autoantigens. Starting with exogenous antigens, again, these come from the outside of the body. Okay, so for example, microorganisms. This looks like a bacterium right here. This might be, not sure, a virus. So these contain antigens on, for example, their cell surface. Okay, this bacterium is actually expressing some sort of what looks like glycogens, and these glycogens, again, are specific for binding to other cells. In this case, host cells. In this case, host cells for bacteria. Those can act as antigens. Okay. This here seems like a protein that is coming out of the bacteria, maybe the flagella. If it's the flagella that we're looking at here, then the flagella of the bacteria can act as an antigen. What our cells can do with the flagella is they can go ahead and break it down into its constituents, take a piece of those and make that an antigen, and then show it to the rest of the immune system, so that antigen presenting cells can go ahead and look for markers that look like these flagella and destroy them. Okay, so same thing with the glycoproteins of this microbe here. This looks like a virus. Viruses are not alive. They don't contain cells. They don't contain a cell surface, although some contain an envelope. But nevertheless, the glycoproteins here, these spikes, act as antigens for that virus. Then we have endogenous antigens, which have an origin inside of the cell. So once a virus, for example, enters a cell, this virus, as you recall, becomes embedded on the host cell's DNA. So after either a few hours or a few days, maybe a few months or years, that virus becomes active, and it becomes a lytic virus, which means, if you recall, that this virus is going to go ahead and start generating its own proteins. It's going to go ahead and start expressing its own proteins using the cell's machinery. Okay, the RNA polymerases for transcription, the cell's ribosomes for translation, and all the cell's organelles to be able to make all the proteins for the virus so that the virus can reproduce. Okay, so those proteins that the virus produces, those can act as antigens as well. Then we have autoantigens, which are not necessarily harmful for us, because we actually recognize these as as natural. Okay, these are innate antigens that we are essentially born with. Then we come to another major term in immunology. Okay, so especially in the adaptive immune system, there's something called the MHC, okay, or major histocompatibility complex. These are almost like cell tags. They're unique identifiers for cells, and there's many different varieties. But let's go ahead and talk about real quick about two classes: Class 1 MHC, Class 2 MHC. Okay, so Class 1 major histocompatibility complexes are located on every single cell that is nucleated. So eukaryotic cells are, they're characterized by the fact that they have a nucleus in their cells. Most of our cells contain a nucleus except erythrocytes. So most of our cells will contain Class 1 MHC molecules on the cell surface to be able to bind antigens. Okay, so that's basically why they're important for immunology, because our cells use major histocompatibility complexes to be able to bind antigens. Okay, so why would that be? Well, because these cells will, aside from all of the functions that they already have, they also want to protect themselves. Okay, so they also want to protect themselves, and they at some point will become antigen presenting cells, so that the rest of the immune system can help them in case they become infected. Okay, so that's what antigen presenting cells do. They present a piece of the antigen right on the outside using their MHCs. So that little antigen is going to go ahead and hang out right there to alert some other cells of the immune system to come and deal with that cell that is infected. There's also a class 2 MHC molecule, and these are located on B cells or other antigen presenting cells, and I'll go ahead and get into the specifics of this in a little bit. Again, what's important here is the fact that these major histocompatibility complexes, they are binding sites for antigens, and ultimately they become unique identifiers for dendritic cells, which we're looking at here. These are dendritic cells. Dendritic cells are also known as antigen presenting cells, or APCs, and they're specific because they contain dendrites. Okay, these dendrites are extensions to the cells. This is one cell right here, and these dendrites are extensions to that cell, to those cells, which contain what is known as arborizing antigens, if you will, or processes, which are used to capture pathogens, to go ahead and bind to other MHCs, and in that manner they manage to deal with pathogens or infected cells. Alrighty, so antigen presenting cells or dendritic cells, they are meant to recognize infected cells. Okay, and then infected cells, their job is to tell APCs, tell antigen presenting cells, that they are infected. Okay, because without these infected cells telling APCs that they're infected, then it's pretty hard for APCs to be able to identify what cells are infected. Okay, that's why major histocompatibility complexes are important, because they allow infected cells to signal APCs that they are in fact infected. Again, there are specific processes that I'm going to talk about later. Let me just kind of show the overview here.
Certain phagocytic cells of the immune system, called antigen-presenting cells, seek out foreign cells and engulf them. In addition, despite the best efforts of the immune system, cells can become infected. In both situations, the cell needs a way to communicate with the immune system cells. The phagocyte needs to show what it's captured. The virus-infected cell needs to show that it's infected. Antigen processing and presentation is a way for a cell to display information about its activities.
So from there, that little antigen right there goes ahead and finds its MHC. It binds to it, and again, that allows for that allows for the immune system to engulf or opsonize or neutralize pathogens. Alright, so a little bit of biology here. This is where biology and microbiology meet, where they merge, right? So that's why biology is important for everything else. It forms the foundation. So in processing endogenous, and for that matter exogenous antigens, the ER, the endoplasmic reticulum, is needed. Recall that there's two types of ER. There's the smooth ER and there's the rough ER. Okay, this is the rough ER right here. its job in processing endogenous antigens, which again, remember that these are basically proteins that were made by bacteria or viruses that managed to get into cells. Okay, so these are infected cells right here, trying to act against the virus or bacteria. Okay, so what they're going to do is in response to the polypeptide, otherwise known as a protein, produced by the bacteria or the virus, what they're going to do is they're going to go ahead and intake those polypeptides into the endoplasmic reticulum of the cell. Okay, so by that point, those polypeptides already became epitopes. Okay, so these are antigens, parts of the antigen. Inside of the ER, they go ahead and bind to class 1 MHC molecules, and from there, they're packaged inside of the Golgi apparatus. And what the Golgi is going to do is it's going to go ahead and send them out using vesicles. Okay, so these vesicles bud out of the Golgi apparatus, and those are parts. The vesicle is essentially a sac-like structure walled off by a plasma membrane. So this would be a vesicle right here. It came from the Golgi apparatus, and it's again surrounded by a membrane. There is nothing inside except for the MHC1 molecule bound to the antigen right there. So from there, last thing is that antigen, along with the MHC, is embedded onto the cell surface or to the cell membrane on its outer surface, and everything from there is basically using other cells of the immune system like macrophages to come over and deal with these infected cells. Alright, so that's endogenous process. Now exogenous processing is very similar, okay? So very, very similar to endogenous processing, but these antigens do not originate from the inside of the cell, they originate from the outside of the cell. So, what we're looking at here is we're looking at a macrophage. Okay, this macrophage engulfed an antigen or a microbe, and by this time, once it's engulfed, then this microbe is gonna go ahead and be degraded inside. It's gonna be broken down into its constituents. Those constituents become epitopes. Okay, they become antigens. But then this time, they're going to go ahead and bind to not MHC1, but MHC2 molecules inside of vesicles, inside of the APC or the macrophage here. And once that happens, then these vesicles go ahead and travel over to the cell surface, to the membrane, and they embed those molecules just like just like in endogenous processing, they embed the molecules right onto the cell surface, so that further cells of the immune system can deal with the infection, and not only that, but also look for other cells that are infected, right, that are showing the same epitopes and the same antigens on their cell surface. So here are more details of what I just mentioned.
Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants. Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow the cells of the immune system to detect proteins inside the cell without entering the cell. Cells in the body routinely break down proteins into small peptides, which are about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol. Vesicles containing the MHC peptide complexes travel through the cytosol to the cytoplasmic membrane, delivering the complexes to the exterior of the cell. If this cell is functioning normally, the immune system cells will simply ignore this presentation. However, if the cell is infected and thus presenting viral proteins, the immune system cells can notice the problem and take action.
So in the case of autoantigens, then that's when the immune system ignores those cells. But again, in the case of an infected cell, whether it's a virus or a bacteria or a protozoa or a fungi that is infecting the cell, then the immune system goes in and takes further steps to deal with those infected cells. And for the most part, they are killed. Those cells are killed. All right. So because there's no other way to deal with with the antigen. Again, two classes of major histocompatibility complexes. There's class 1 and class 2. Class 1 is used for endogenous antigen processing. Class 2 for exogenous antigen processing. Most of your cells in your body act as class 1 antigen presenting cells. Okay, so
antigen-presenting cells, such as macrophages and dendritic cells, have two classes of special proteins that are used to present antigens on their surfaces. Collectively, these proteins are called MHC, which stands for Major Histocompatibility Complex. Class 1 MHC proteins are used to present antigens produced within the cell. A different class of MHC proteins, Class 2 MHC proteins, are used to present antigens engulfed by antigen-presenting cells. Most of the other cells in the body, including infected cells, are also able to present antigens. However, these cells only have the Class 1 MHC proteins.
So here is what I'm showing here is a T cell receptor. Okay, keep in mind T cells or T lymphocytes, they are produced in the bone marrow. Excuse me, and they migrate to the thymus. From the thymus, they are matured within the thymus gland, and then they go ahead and move to the rest of the body through means of the lymphatic system, through lymphatic vessels, lymph nodes, spleen, Peyer's patches. So once in the body, what they do is they're gonna go ahead and essentially just wait for things to bind to. Okay, so these receptors are meant to bind to antigens, and these antigens must be found associated to major histocompatibility complexes. Okay, so in other words, these cannot bind to pathogens themselves. They need to bind specifically to pathogens that have been processed exogenously or endogenously by antigen presenting cells, and they must be found on the cell surface or membrane of the antigen presenting cell. Okay, so in that manner, these can go ahead and bind to each other. Those two can bind to each other, and the processing of that cell, the infected cell, can take place after that. So these are several types of T cells or T lymphocytes. We have T helper cells of the 1 type or of the 2 type. We have cytotoxic T cells and also regulatory T cells. All of them are represented by glycoproteins on their cell surface, which are known as CD4, the T cell receptor, CD8, CD95, and CD25. Okay, CD4 specifically is associated with HIV. Okay, HIV utilizes the CD4 pathway to be able to bind on to helper T cells. Okay, and render them useless ultimately. All right, so now last thing from here is the secretions of these cells. Secretions range from interleukins to perforins, granzymes, cytokines, and all of these would be signaling molecules which are going to go ahead and signal the rest of the immune system to go ahead and help out, or maybe induce clonal selection, so that the immune system can be completed, or immune response can be completed. So what I'm showing here is a type of clonal selection which is undergone by helper T cells. Again, clonal selection is a production of many clones, many copies of the same cell, and these clones are going to be identical in every single aspect to the original cell. In this case, these are helper T cells, which are helping in create memory T cells. So about half of them are going to go ahead and become memory T cells for future infection, and then most of them, or the other half, are going to go ahead and become mature T cells to be able to deal readily, right? So be readily available for infection that might happen right away. Okay, so now keep in mind that T cells, helper T cells, are gonna express a receptor called the T cell receptor, and that they're gonna go ahead and work by means of the CD4 receptor, which is gonna go ahead and bind to a major histocompatibility complex found on certain antigen presenting cells. So once that happens, then interleukin 1, which is a certain kind of cytokine or a signaling immune molecule, and that's going to go ahead and essentially activate the clonal selection and the proliferation of these cells to produce, again, the two types, memory cells and mature T cells.
Each helper T cell produces a unique T cell receptor, or TCR, which corresponds to a specific antigen. Each helper T cell also displays a receptor called CD4 on its surface, which recognizes the MHC molecules of antigen-presenting cells. When the helper T cell encounters an antigen-presenting cell with that antigen displayed in an MHC molecule, it signals the antigen-presenting cell to release interleukin 1. The IL-1 stimulates the helper T cell to produce IL-2, as well as IL-2 receptors. The helper T cell can then auto-stimulate itself, keeping it active as it proliferates. Once a helper T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these helper T cells differentiate into long-lived memory T cells, but most mature to produce cytokines to help with the immune response. Helper T cells differentiate into two subpopulations called Th1 and Th2. Th1 cells produce cytokines that primarily activate the cells of cell-mediated immunity, such as macrophages, cytotoxic T cells, and natural killer cells. Th2 cells produce cytokines that activate the B cells involved in humoral immunity.
That last part is very important, okay, because these two are, the two different branches of the adaptive immunity, our adaptive immune system, and they are activated by these two subtypes of helper T cells, 1 and 2. Okay, so cell-mediated immunity is going to go ahead and utilize cytotoxic T cells to go ahead and attack infected cells. It's going to go ahead and stimulate macrophages, okay, which are going to work by phagocytizing or engulfing infected cells. And then natural killer cells are going to go ahead and act against virally infected cells, but also cancer cells or tumor cells. Alright, so now the humoral immunity is going to go ahead and utilize B cells and produce antibodies to be able to either opsonize or neutralize pathogens. Now for cell-mediated immunity, is we have B cells rather, B lymphocytes. Alright, so we're looking at the receptor of B lymphocytes, which is very specific for antigens. These receptors cells, B cells, are going to produce 5 different types of antibodies. Right, so antibodies are also known as immunoglobulins or IgGs, and there's 5 different kinds. There's IgG, there's IgA, IgM, IgE, IgD. And these 5 major types of antibodies will go ahead and act against pathogens directly by binding to them, surrounding them, and then signaling antigen presenting cells for their destruction. All right, so again, we're looking at a B cell right here with its B cell receptor, or BCR, and then the antibodies that it's producing. This Y-shaped structure right here is an antibody. Here's another look at a B cell. It's also known as a plasma cell, and this is using a transmission electron microscope. So this is not an animation. This is an actual cell by electron microscope view. So again, what we're looking at here is we're looking at the nucleus of the cell, and this is the entire cell right here. Most of it is made up of rough endoplasmic reticulum. It's got a lot of Golgi bodies as well. Keep in mind, these are specific for the production of antibodies. 5 different kinds of antibodies. Antibodies are proteins. Okay, so therefore the importance of having such an extensive set of endoplasmic reticulum, this collectively is called the endomembrane system. So having such a large endomembrane system is important for protein production. Okay, so which is happening at all times in these cells. So we have 5 different types of antibodies which have different functions here. They all kill pathogens, and they do so in different ways. First is neutralization. So by neutralizing, meaning that antibodies are going to go ahead and bind to either the virus itself or the proteins or toxins that the virus is producing or the bacterium is producing, and in that manner they go ahead and neutralize either the toxin itself, protein, or bacterium, or the virus itself. Okay, so that's one. Second is opsonization. What opsonization is going to do is it's going to go ahead and utilize antigen presenting cells or dendritic cells to be able to engulf these microbes which were bound to antibodies. So you're looking at a microbe right there filled with antibodies. Those antibodies went ahead and signaled an antigen presenting cell here, or maybe a phagocyte, right? And that phagocyte went ahead and, like a macrophage for example, went ahead and engulfed that entire complex right there. Third is by agglutination. Okay, so antibodies also work by agglutinating molecules, which means aggregating them, collecting them in clumps, making clumps out of them. And then next is oxidation. So creating these ROS, or reactive oxygen species. These cells essentially die because of oxidation. Okay, those oxygen species are harmful for all cells, our cells and bacterial cells. Then last but not least is by antibody-dependent cell-mediated cytotoxicity. Cytotoxicity referring to cyto means cell, toxicity, well, you know what that is. So something that is toxic to the cell. Okay, so these, for example, natural killer cells here are going to go ahead and induce cytotoxicity within the microbe by using these proteins called perforins. And then further, they're going to go ahead and make a hole, right? So these perforins are specialized to make pores on the cell surface of the microbe. So from there, the natural killer cell injects what's known as granzyme. Right? Granzyme enters the microbe and induces all of these apoptotic proteins inside of the microbe, which causes apoptosis. Apoptosis is programmed cell death.
The antibodies produced by plasma cells have several functions. Since each antibody has at least 2 antigen binding sites, antibodies can bind to more than one microbial cell, resulting in agglutination or clumping. This agglutination helps remove the microorganisms from the body. Antibodies can also neutralize toxins, viruses, and bacteria by binding to critical portions of the toxin or blocking proteins necessary for attachment to cells. Antibodies also act as opsonins for phagocytosis, and activate the complement system.
So here's the activation of clonal selection on cytotoxic T cells, which begins by antigen presentation. So here's an APC, or DC stands for dendritic cell, which is an APC, or an antigen presenting cell. So these APCs are going to go ahead and bind to helper T cells. Okay, so these helper T cells and that binding will cause the dendritic cell to release interleukin 12. Okay, so this interleukin 12 will go ahead and help the helper T cell differentiate into a helper T1 cell and a helper T2 cell. Alright, so you got two different types. Now, what the helper 1 T cell is going to do is it's going to go ahead and, by the production of interleukin 2, it's going to go ahead and cause clonal selection of T cells, which means those T cells are going to go ahead and make a lot of copies of themselves to be able to form memory, but also to stay activated here and be able to fight off bacteria right away. So this is happening essentially at all times. These memory T cells are not necessarily active at that particular time, that particular point in time, but they will become active later on in future infections. Okay, now you have a cytotoxic T cell, which is bound to a cell that became infected with a virus here. As you notice, there are different structures that are helping in the binding of these two cells, or the binding of the active cytotoxic T cell to the infected cell, to include the T cell receptor right there, the CD8 receptor right here, the epitope of the virus, the major histocompatibility complex of the class 1. So this would be right here an antigen presenting cell, which was again infected by a virus, and it's presenting itself to a cytotoxic T cell for its own destruction.
Each cytotoxic T cell produces a unique T cell receptor, which corresponds to a specific antigen. Each cytotoxic T cell also produces a receptor called CD8, which recognizes the MHC molecules of infected cells. When a cytotoxic T cell encounters an infected cell with a specific antigen bound in an MHC molecule, the cytotoxic T cell produces receptors for interleukin 2 and other cytokines, such as gamma interferon, IFN gamma. The cytotoxic T cell can then be activated by cytokines from activated helper T cells. Once a cytotoxic T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these cytotoxic T cells differentiate into long-lived memory T cells, but most mature to attack and destroy infected cells. Cytotoxic T cells can destroy infected cells through multiple pathways. The best understood of these pathways is the perforin granzyme cytotoxic pathway. When the cytotoxic T cell encounters an infected cell with a target antigen bound in an MHC molecule, the T cell secretes two types of protein molecules, perforin and granzyme. The perforin forms a pore in the membrane of the target cell, allowing granzyme to enter the cell. The granzyme activates the enzymes inside the cell, leading to programmed cell death or apoptosis.
Alright, so a few things to address here real quick. First and foremost, INF stands for interferons. Let me go back to where it was. Let's see if I find right there. Yeah, so INFs are interferons. Okay, these interferons are highly specific. They're very specific for viruses. Okay, they don't do anything else, but they go ahead and signal infected cells that have been that have viruses inside of them. Okay, so these viruses are causing the cell to produce interferons that are signaling to helper T cells, and essentially telling them, what it is that they're dealing with and what it is that they need to be looking for. So this specific virus caused the binding of the cytotoxic T cell by using the CD8 receptor. But in the case of HIV uses CD4, okay, CD4 helper T cells, which renders them useless essentially. So if our helper T cells are useless, then most of our cell-mediated, therefore antibody-mediated immune responses are, they're not of a lot of use. Yeah, so we still have our innate immune response. Yeah, so or at least in HIV cases and other cases as well. So those patients, they don't necessarily use T cells or cell-mediated immunity or antibody-mediated immunity, but they do have their innate immunity and their surface barriers and whatnot to be able to fight off infection. But again, those are not enough. Okay, we rely mostly on adaptive immunity to be able to fight off pathogens that get into the body. Now, here is another way in which cytotoxic T cells can work is by using the CDL95 or CD95 rather receptor here. It's basically the same thing as the other pathways, CD8, CD4, but in this case, what's going to happen is two things. First is a perforin complex is going to be made, or a pore, like I mentioned earlier, is going to be made, and then granzymes are going to go ahead and enter the infected cell. So that's going to cause the infected cell to activate enzymes, as was mentioned, enzymes which are going to go ahead and induce apoptosis. So this cell will ultimately kill itself. Okay, so apoptosis is programmed cell death. So now T cells don't necessarily produce antibodies themselves, but they can cause B cell activation, which produces antibodies. Okay, so that's what you're looking at here. These are T cells that are basically activating B cells so that they can produce antibodies themselves. Okay, so let's go and start off with the antigen presenting cell itself, or the dendritic cell, presenting to a helper T cell. So what the helper T cell is going to do is, just like in the other process I showed, it's going to go ahead and differentiate right into Th1 and Th2. So this Th2, through the use of interleukins, right, it's going to go ahead and activate B cells, and then these B cells, they're going to start cloning, right, clonal selection, and then once they are mature, then they're going to go ahead and produce antibodies, and those antibodies are going to go ahead and neutralize or opsonize pathogens. So through the use of a T-dependent antibody immunity, B cells are going to be able to produce their B cell receptor. These B cell receptors are going to go ahead and have the ability of binding to the cell surface of bacteria, to viruses as well, and in that manner cause clonal selection and opsonization, neutralization of pathogens.
The process of B cell activation is called clonal selection. During this response, a B cell with a specific antigen receptor recognizes an antigen. This cell proliferates to produce a multitude of cells, also called a clone of cells, with the same specificity, which differentiate into memory B cells and plasma cells. This process is called clonal expansion. The antibodies produced by the plasma cells bind to antigens to produce the humoral response. In the simplest form of B cell activation, an antigen called a T-independent antigen enters the lymphatic tissue and binds to a B-cell receptor, or BCR, on the surface of the B-cell. The B-cell receptors on a particular B-cell are identical and very specific to just one antigen. Once the BCR binds to the T-independent antigen, the B-cell becomes activated. It's now ready to proliferate. However, most antigens are not potent enough to activate B-cells by themselves. In these cases, the B-cell will require a second opinion from a helper T-cell in order to become activated. We call these antigens T-dependent antigens. The helper T-cell recognizes the antigen as it's presented by the B-cell and secretes cytokines to tell the B-cell to become activated. Once a B-cell becomes activated, it proliferates or divides, forming a clone of cells specific to the same antigen. While some of these B-cells differentiate into long-lived memory B-cells, most differentiate into plasma cells, which secrete antibodies against the antigen that activated them. The antibodies can enter the bloodstream and travel throughout the body. Plasma cells only live for a few days, but each one can produce about 2,000 antibodies every second, resulting in enormous quantities of circulating antibodies.
So many STDs work in this matter. Okay, so in this fashion rather. These processes can take anywhere from days to months, to weeks to months to years to be able to produce these antibodies. In the case of HIV, for example, HIV takes years. Yeah, so not only to start showing symptoms, but years for the patient to even show minimal amounts of antibodies collected in their tissues, especially because HIV kills helper T cells. Okay, so that in and of itself basically gets rid of the antibody production, antibody generating mechanisms. But other STDs, like for example herpes of the type 1 or type 2, or HPV or molluscum contagiosum, those take months. Yeah, so those take months. Sometimes in some patients to be cleared from their bodies or the lesions that they cause. In the case of molluscum or herpes simplex 2, they cause lesions in the pubic region, the genital area. Those lesions, again, they don't go away quickly. They take sometimes even months or up to 3-4 years, according to some studies that I've read. So that is how long this process can be in some diseases. Other pathogens, like for example within the influenza family of viruses, well, they can take weeks, right? So that's a faster response. They take weeks, and within a matter of 1 to 2 weeks, then the person, after infection, the person is already better. But again, we are exposed to those viruses ever since we are little. Yeah, so say for example somebody is barely infected for the first time. Yeah, so they show a primary immune response, which takes, in this case 15 days for IgG or immunoglobulin G or antibody G to be able to be made. And 15 days would be the incubation period, for example, of SARS-CoV-2, anywhere from day 3 to day 15, or COVID-19. But then, or any influenza virus. But other viruses take, again, up to weeks, months, sometimes years, for the primary response. So this would be the first time that the person is infected by that virus. Now, a secondary immune response takes less time. Well, why would that be? Well, because that person has already been exposed to the virus before. So therefore, within 3 days of infection, they're going to go ahead and have a high amount of IgG Okay, keep in mind that these were immature. They weren't even, sometimes they weren't even there, but, or at least in the primary response they were not there, but because the person was exposed to the virus already at least one time, then they were able to form enough memory cells, memory B cells, so that those B cells in the secondary response, they can skip essentially all other steps and goes straight to producing the antibodies that they're made to produce. So in this case, that person, which is the same person there, is not gonna show symptoms for very long. So if anything, 3 days, 4 days, instead of the 15 days, 20 days, 25 days after infection.